So now let's look at different types of cells. Here you can see a prokaryotic cell, bacterial cell, and here's a eukaryotic cell, very large comparatively. Typically we assume prokaryotic cells are smaller, they are simpler in structure. They have DNA, but their DNA is usually one single chromosome, and it is a circular chromosome found in a region called the nucleoid. They have ribosomes, but they don't have other types of organelles. Eukaryotic organisms tend to be larger. They have more complex structures. Their nucleus is where they hold their DNA. They contain multiple organelles and ribosomes. Do you have a cute song to kind of start off this video? I like the song. It's always fun to play songs. So. I am a prokaryote, a simple little cell. Much inside me, it does me very well. Cell membranes and ribosomes and scattered DNA. Just a necessities, and I do okay. Well, sometimes I have a flagellum. Otherwise, I'm quite simple. Yes, that's called the nucleoid. But that's floating around in the called cytoplasm. Did I hear say that right?
Let me check my script. Oh, yes, it's that important. Just remember that I'm really simple. But despite this, I'm capable of withstanding extreme conditions, which is quite an advantage to me as compared to my cousin, the eukaryote. Speaking of whom, here it comes. Hi, cousin. Hiya. Here we go. I am the eukaryote, much more than one. I've got all my organelles, which makes me quite fun. They have many functions, but their list is really long. Oh, well, to find out, you'll have to wait for the next song. Wait for the next song.
Boy. Oh, wait, hold on. I love that. Oi. Okay, sorry. Anyways, let's go back to the video. All right, so I just did that. I'm sorry. I sometimes make mistakes. Well, which is not unusual. Oh, wow. I'm having trouble with this, I think. Okay. And there we go. All right, so let's talk about prokaryotic cells, and then we'll talk about eukaryotes. So prokaryotic cell structures. The term pro means pre, and the term karyote means nucleus. So a prokaryotic cell is a pre-nucleus cell. It has no nucleus. All prokaryotes lack a membrane-bound nucleus. Prokaryotic organisms, we typically say they have no organelles. They are unicellular, but they can be found in forming like clusters or strings, or they can even be found in multicellular associations like biofilms. The prokaryotic organisms live in either eubacteria or archaea. Those are the domains. And the organisms we typically think of when we're talking about these are bacteria. So bacteria are prokaryotes, but they do not always cause disease. So archaeobacteria have never been found to cause actual disease. Eubacteria can cause diseases. and some of those diseases are really bad, but the majority are not disease causing. So we have more bacterial cells on our body than we have human cells, and we have trillions of human cells, which means we have lots more bacterial cells. So trillions times trillions times trillions, think of that. And those bacterial cells make us healthy. So they're typically very beneficial. Bacteria can be found in almost any environment. So all environments that eukaryotic organisms can live in, plus some extreme environments that eukaryotes can't live in, such as hot springs, volcanoes, hydrothermal vents, the Dead Sea. This is bacteria. Bacterial cells have a region called the nucleoid where they keep all their DNA. They have tiny little ribosomes found throughout. They have a plasma membrane. All cells have a plasma membrane. They have cell walls, and many have a glycocalyx cover called a capsule or a slime layer. Some bacteria also contain flagella. So this is a single flagellum. Some bacteria can contain multiple flagella surrounding them. And many bacteria contain fimbriae. Fimbriae, or pili, depending on what you want to call them, are tiny structures that either allow bacteria to adhere to surfaces or adhere to other bacterial cells, depending on the type. Eukaryote then, eu means true, karyote still means nucleus, which means eukaryotes have a nucleus. We have a nucleus. We typically are very complex. We are all found in the domain eukarya. We have organelles, multiple organelles, and we can be single-celled or multicellular. So animals, which is what we fall under, plants, fungi, and protists are all eukaryotic organisms. Cell walls in organisms that have them, they function to help maintain the cell shape and integrity, especially due to osmotic changes. They protect the cell. So plants have a cell wall, fungi have a cell wall, algae have cell walls. The carbohydrate that is found within the cell walls is dependent on the type. So in plants, we have cellulose. In fungi, typically we see chitin. And chitin is really strong. So chitin is what also makes up the exoskeleton of insects. This is a plant cell wall, or a plant cell, not cell wall. You can see the cell wall out here. The plasma membrane is here. And then inside we have all the organelles as well as the nucleus with the DNA. Here's an animal cell. Animals do not have a cell wall, so here's the plasma membrane. Inside we have all the organelles and the nucleus. So the cells have three main structures, or regions, I guess, not structures. We have the plasma membrane, which is the outer region that functions just like bacterial plasma membranes well, like all plasma membranes. They are selectively permeable. Then inside we have the cytoplasm, which contains all the organelles, so that's termed a cytosol. And then we have the nucleus. So let's talk about these three structures. The nucleus is the site of the cell that we typically think of as the brain. So it stores our DNA, and DNA is the blueprints of who you are. Every single one of your cells has a complete set of your DNA. Not all the DNA is going to be activated in every single cell, but every cell does have a complete set of your DNA. So it's considered the control center. The nucleus has three basic structures associated with it. The nuclear membrane, which is a double membrane that has these little tiny pores with it. The chromatin that's found within the nucleus, and then in the center, most nuclei have at least one, and some have more than one nucleolus, which is a structure inside of the nucleus, and that's where RNA is formed. So the nuclear membrane is a barrier. It's a double membrane. It has two plasma membranes, and it has pores that allow for nutrients to move in and materials to leave the cell. The nucleolus is the site of RNA ribosome production. And so once the ribosomes are produced, so ribosomes are composed of RNA, they leave the nucleus, they enter the cytoplasm, and then they can either just stay free-floating in the cytoplasm, or they can attach to the endoplasmic reticulum, forming a rough ER. The nucleus has chromatin. Chromatin is DNA that's unbound. So DNA, when it's condensed, is known as a chromosome. But as an uncondensed form, it's just free, scattered inside of the nucleus, and then it's called chromatin. So each organism has its own set of chromosomal numbers. Humans have 46 chromosomes inside of each of our nuclei. Dogs have 78 chromosomes. Peas have 14 chromosomes. So each different organism has a different number of chromosomes. Here you can see the DNA a little bit better. You see the nucleolus and then that double membrane. The cytoplasm then is outside of the nucleus but inside the plasma membrane, and it's filled with all of the organelles. One of those structures is called a ribosome. So ribosomes are double or two-part structures. They consist of a small and large subunit. The large subunit is known as a 60S subunit, and the small one is known as a 40S subunit. They make up what we call 80S ribosomes. They are either going to be found embedded in the rough endoplasmic reticulum, or they'll be just free-floating in the cytoplasm. Their function is for protein synthesis. That's what they do. So attached to the nucleus, we have an endoplasmic reticulum. The endoplasmic reticulum is a series of tubes that connect from the nuclear membrane, or from the nucleus, I should say, and run out to the Golgi. They don't attach to the Golgi, but they run out to the Golgi. There are two types of endoplasmic reticulum, rough and smooth. Rough is studded with ribosomes, smooth is not. So rough endoplasmic reticulum functions to produce proteins. The ribosomes itself produce the proteins, and then the endoplasmic reticulum modifies the proteins and transports them to the Golgi. The smooth ER produces lipids. So its function is to make up steroid molecules like cholesterol. It breaks down carbs. It can store certain molecules like calcium ions. It functions in detoxification, so it breaks down toxic chemicals. And so you'll see more smooth ER in regions like the liver, where we have, toxic breakdown all the time. So our liver is kind of our protector of the body. And so here you see the rough ER, and then that leads out to the smooth ER, and the smooth ER next to that is typically the Golgi, which is right there. So you see smooth ER to the Golgi. The Golgi apparatus is kind of like the post office of the cell. It takes in the proteins produced on the rough ER, and then it packages those proteins and sends them out to the different areas. It can send these proteins to the plasma membrane to be embedded in the plasma membrane. It can send them to the plasma membrane to be excreted by the cell, or it can release them directly into the cell, and those are vesicles like lysosomes and peroxisomes. So here we have the rough ER, and here's the Golgi. So proteins come from the rough ER, they enter the Golgi, and they move, and then depending on the function, they're either going to, a portion of the endoplasmic reticulum will bleb, or a portion of the Golgi will bleb off, forming a vesicle. That vesicle was attached to the plasma membrane and excrete the proteins out. Or the vesicle that is produced from the blebbing off of the Golgi will attach to the plasma membrane and become part of that plasma membrane, and the proteins will stay right there. Or we might just bleb that vesicle off, and now we have a new cell organelle like a lysosome. The lysosomes are structures, are vesicles filled with digestive enzymes. They break down proteins and DNA, and RNA. They're basically the digestive system of the cell. They break down structures when materials come in that the cell doesn't want. The cell will bind the material with a lysosome, and the lysosome will break down everything. Peroxisomes are structures very similar to lysosomes in shape, but they're smaller than the vesicles or than lysosomal vesicles. Peroxisomes also have they have enzymes inside of them. These enzymes tend to break down molecules and produce a byproduct called hydrogen peroxide. So they use oxidation processes. And then hydrogen peroxide, which is also toxic to our cells, can be broken down by other enzymes inside of the peroxisome to water and oxygen, which is not harmful to our cells. Vacuoles are organelles inside of cells that can contain different nutrients or water, water is a nutrient, so I guess it would work both ways. The most common cell structures, eukaryotic cell structures that have vacuoles are plants. And vacuoles tend to hold a lot of extra water in for those time periods when water is lacking in the environment. So talking about plants, plants have chloroplasts. Chloroplasts are the site of photosynthesis. Photosynthesis is the production of carbohydrates from sunlight energy. So we take in sunlight energy and we convert it to energy of a carbohydrate. Chloroplasts are double membranes, or contain a double membrane. They have their own circular DNA, very similar to bacterial DNA. They have ribosomes that are similar to bacterial ribosomes, so they're 70S ribosomes. And inside the chloroplast, they have these stacks of thylakoids. And when we have a stack of thylakoids, we call it a granite. It looks kind of like a big stack of pancakes. and this is filled with chlorophyll, and that's the light attracting pigment. So here you can see the chloroplast. These are the thylakoid membranes that are stacked in grana. The outside of the, or I will say inside of the chloroplast, outside of the thylakoid, is the stroma. And so we have light-dependent and light-independent reactions occurring in different areas. The mitochondria then is the site of aerobic cell respiration. All eukaryotic cells have mitochondria. Chloroplasts are only found in plants and algae, but all cells have mitochondria. So they're the energy production cells. So like the AMER and IP, I guess, if you want to think of it that way. They also have a double membrane. The inner membrane is known as the cristae. The matrix is found inside the inner membrane, and this is where aerobic respiration occurs. So it kind of looks like a kidney bean. And so here's the outer membrane, here's the inner membrane. The inner membrane is kind of, it forms all these different invaginations in the matrix. The cristae here is where we have what we call an electron transport chain. Inside here we have other functions for aerobic respiration. The mitochondria also has its own DNA, 70S ribosomes, and double membrane I said. So organelles, the chloroplasts and the mitochondria function together. Chloroplasts take in sunlight energy and produce carbohydrates. Those carbohydrates can be broken down in the mitochondria to produce ATP energy, which is usable by all cells. These two organelles are also important in determining endosymbiotic theory, how we produced multicellular organisms and eukaryotic cells, which is really important, from single-celled bacteria. Normally I would stop here because I've already went for 22 minutes, but we only have a few slides left, so I'm just going to finish this up on this video. So the cytoskeleton is our last organelle that we'll talk about, and the cytoskeleton functions in maintaining the cell shape. It functions in moving organelles to proper positions. It moves other substances within the cell, and it functions in the movement of DNA during mitosis and meiosis. There are three subunits to, or three components to the cytoskeleton. We have actin filaments, intermediate filaments, and microtubules. So microtubules are the largest, and then we have these intermediate filaments and then actin. Altogether, these form the cytoskeleton, and the cytoskeleton forms other structures as well. So microtubules form the centrioles within the cell. Centrioles are what control the spindle fibers so they form these spindle fibers that control DNA movement during cell division. And microtubules also function in movement of entire cells. So they form cilia and flagella. Cilia, oh yeah, here's cilia. Cilia tend to, they're like hair-like or finger-like substance or structures, sorry. And they will either move materials along the cell surface, or they'll move the entire cell. A lot of times ciliated cells tend to move materials towards the mouth opening or towards the opening of a lumen. Flagella are found on cells, and they function in, wow, that was loud, wasn't it? Sorry. They function in movement of the entire cell as a whole. Sorry. So here we have cilia. These are small, typically, kind of like finger-like structures, and they can move materials along the cell. They tend to not move entire cells around, but more move materials along the surface of the cell. Ciliated cells can move cells probably very short distances, but typically flagella work better. And then we have flagella. Flagella are long structures, more like hair-like structures. Typically, you see flagella as single, whereas in bacteria, when we have flagella, they can be multiflagellated. These move cells along distances through, you know, like this is a sperm, so the sperm would move through the vagina, cervix, the entire uterus, all the way into the fallopian tube to try and get to the egg where it could fertilize the egg. That would be an example of flagella working. That's the end of this chapter, so we're going to be talking about this next week. This week, don't forget, we're going to be talking about chapter 2.